I'd like to introduce Dr. Clifton Haider. I have known Clifton for a very long time now. Clifton entered the graduate school well over a decade ago and immediately had a huge impact in the field of MR imaging. And when I say huge impact, I mean recognized around the world, implemented in MR scanners around the world. Right? Yep. But today, Clifton's going to talk about some of his other work, and this work is, as you can see, in physiologic monitors. The whole space of wearables, home healthcare monitoring is really exploding and will have a huge impact on healthcare going forward. And some of the work that Clifton's going to talk about is literally a decade ahead of what's going on out there in industry, and so we're really excited. He's going to speak for about 40-45 minutes, and then he's going to take some questions, and then we're going to go on and watch an extreme sports movie. So thank you all for attending. Clifton, it's up to you.
So I want to cover some work we did a number of years ago, and it's interesting and kind of relates to the program for today because it was a Mount Everest expedition, and it was fielding a device that could run for an extremely long time on a person without them interacting with it, recording data 24/7 on a mountain in an extreme environment. But what's also interesting is that the fundamentals of that engineering were done fairly rigorously so that it's actually continued on, and we're still using it 5 years later. We're still writing new algorithms that can run on that hardware, and the data we're getting with it is still being used, and we're using it for new collaborators, new sponsors across the United States. And so I think it was a very interesting effort, and I wanted to provide some background, general physiologic monitoring, some of the effort. Let's see if we can now hear me. On calibration and how we're moving forward from the original 2012 Mount Everest expedition. And then at the end, for the students, I have a small section of kind of my reflections on things I wish I knew. So as we look at healthcare right now, I think that the majority of the focus is on disease management and detection and treatment strategies. Once something has become intolerable, you go to the clinic, they diagnose it, they start treatment. What I think we should be looking at is more a preemptive strategy. Get out there, look for something pre-symptomatic. And we're looking for technologies that have a very mobile base. So it's hard to move an MR scanner around to every school, and it's unlikely that parents would let you put an MR scanner in the school to image children. So maybe if they could look at something off their phone that they're already carrying, or some other small device that's minimally invasive or non-invasive totally, that can just co-locate with their books, would be a great thing, as an example. But I think what's also important is that we look at what the NIH has been looking at for some time. And it's really this, it already hurts so much that I have to go to the doctor, and we have a cost that's insurmountable, and it just continues to explode. And as we start getting to early symptomatic, maybe less complex care is required to resolve the issue. Maybe it hasn't spread to another organ. Maybe your joint hasn't deteriorated such that both joints are now needing replacement. And as we get down, so it's preemptive. Hopefully this is showing up. It's preemptive. Maybe we could put some sort of regenerative medicine. We can inject some stem cells into your knee, and it wouldn't have deteriorated to the point where you needed knee replacement, and therefore you wouldn't have had all the complications that go with, or comorbidities that go with advanced stage disease and the complex care and cost. And so that's the premise for physiologic monitoring or remote monitoring. And I think it's unique in its ability to continuously interrogate the body. We don't want snapshots that are very specific but don't really tell us what's going on in the rest of somebody's life. So we have a resting heart rate that we get when we see our physician, but maybe we should be looking at what's happening when they're on the treadmill, at home, or they're out in the yard mowing. How is their heart acting, and other variables. So we want to be able to supplement a snapshot that happens when you visit your physician with continuous data and see if we can begin to build a better picture of what somebody's actual health is and get on the leading edge of the decline so we can do a better job of treating. Again, it gets us outside of the clinic. It's very expensive to come into the clinic when it's symptomatic. Get it outside. Maybe we can demonstrate measures that are useful. And I think what's also exciting is that we're just beginning to see new types of sensors evolve every week you see a new publication on a new optics component that's maybe more efficient or more adept to a measurement that you would like, and you pick your field. Everything's advancing very rapidly, and I think it's nice to see translation to the practice. What's also nice and critical is that to have a mission, you have to have money. And so the NIH, not only have they stated that it's important to start looking preemptively, they're beginning to fund it, and they're beginning to recognize that you can't just focus on genomics, or you can't just focus on regenerative medicine. You have to look holistic across all of your tools and begin to fund individual components. So again, can we look at mobile devices, and can they have an impact on the clinical trials that are going on? Is there a metric that's objective that lets you decide if a drug is working earlier or a drug is not effective or the dose is wrong? All these things are very important, and money does drive ultimately where you're going to go. Another thing is consider whether or not you're going to be regulated. So would you want to determine whether or not you should take an insulin shot based on a photo from your phone that wasn't regulated? I don't think I would. Would I want to interpret my own ECG? Probably not. But there's going to be limits to how much a person is willing to take on and how much the government is willing to let companies let people take on. And so I think considering regulation up front, are you looking for a clinical device, a clinical grade device where actionable data will go to a physician that can affect your health care? Or do you want somebody to maybe improve the number of or amount of activity they do per day to have an effect on their cardiovascular health? And so this is an important consideration, as well as what you claim, and those are intimately coupled. So if you make a claim and it's not FDA regulated, and it proves out in the popular press that it didn't work the way everyone thought it would, there's likely to be a lawsuit or otherwise. And so I think it's important that we consider these up front when we're looking at devices. And more importantly, a lot of the big experts in the field are starting to recognize and kind of put position papers in, which states, here's what our growing perspective or a bottom line should be for a device, and it begins to build a vernacular or some sort of description of what people expect when they put on a device. And I think this is important because there's immense scientific interest in monitoring, and people need to have references that they can go back to and point to and say, we meet the expectations of the experts in the field, and here's how we do that. And I think as we look at Mayo Clinic, we're trying to really deliver real-time patient care to the patient based on evidence-based medicine. And I think that's a real important note versus trying to give something to somebody to anecdotally help their day. And what I want to do is also give some background. Many of you have probably heard the history of Earl Wood and how the development of the G-suit took place in the early 30s and 40s at Mayo Clinic. If you haven't, I'll just give a brief review. So in the late 1930s, pilots were crashing perfectly good aircraft and dying, and the Army Air Corps, which was pre-Air Force, thought the pilot was the cause of it. And so they wanted to understand why this was happening. Mayo stood up the medical sciences building, and for $1, they ended up developing the G-suit. And to do that, they built a human centrifuge. They'd spin people in a quarter turn up to 8, 9 Gs. And you can see someone under an extreme stress. This is Dr. Earl Wood going from 1 G to 8 Gs. And the miraculous thing was the G-suit and this device here were all developed, and this was an incredible race. And at the same time, a quantitative oximeter was developed, and this was called the Wood-Sturm oximeter. And the Luftwaffe, which was the German equivalent of the U.S. military, was desperately trying to do this, but they didn't have a great idea on how to calibrate, and so they went for a different kind of calibration using temperature, which causes the vessels in your skin to dilate to get off the extra heat, and they ended up with big burn marks on their head as opposed to a self-calibrating oximeter. And this work, of course, led to this G-suit, which was more or less the one used in the SR-71 Blackbird, and then a less aggressive suit was used and is used today because of the discomfort associated with the blood being returned back to the central circulatory system so the heart can pump it to your brain. Essentially, they were trying to make humans into dogs because dogs lay flat, and if if you look up here on the top, there's a top rack where Earl Wood predicted that the optimal geometry for a pilot was to be laying flat in the aircraft so that their heart was at the same level as their head. And so if you learn physiology, some of these engineering aspects, when you get to working with humans, might become obvious. One of the things Earl Wood did really well was calibrate, and that's going to be a lot of what my talk is about. So they write the description of their circuit up here, and then he begins to take the devices, the Wood-Sturm oximeters, and he calibrates every single device across all the expected conditions. And then they go out and they capture reference data. So they take an oximetry experiment and they measure the device, and then they measure with chemistry, analytical chemistry, the exact reference value. And they do this across all the expected conditions. If you look at how modern pulse oximeters are calibrated, which are based on the Wood-Sturm oximeter, they're all done in the exact same fashion. However, Earl Wood did it to an extreme that could never be repeated today with people at incredibly low saturations. And again, I just think this is a great slide to demonstrate how good engineering can be done and is done at medical centers. And I think as we look forward, one of Dr. Wood's students was Barry Gilbert. He's the director of the Special Purpose Processor Development Group, and this kind of engineering has continued on today. So these are types of radios, and actually there's a number of SPPDG engineers in here who played instrumental role and actually laid the transistors down on these. So big thanks out to them for the slide. But these are complete radios, and this is not a blown up penny. This was really a really tiny transceiver designed to be carried by an American honeybee so that it could look for outgassings of plastic explosives so that you could clear a field of landmines. And so bees have great senses of smell, and the goal was to develop a radio that they could carry and then locate the bees over the plastic explosives and then save people from having their limbs blown off when they walked across the field. And so again, very well calibrated devices demonstrated to work, and incredible engineering going on at Mayo Clinic. And so the takeaway points from this background is Mayo's had a long history of doing real biomedical engineering, and it still continues today. Often, and I'll agree that sometimes a key technology is developed and then applied to other clinical needs, but usually you have a technical need that then you find your key technology and you have a useful outcome and you iterate, and sometimes you find that you've developed something so interesting that it can apply across fields, and that's fantastic, and you can do tech transfer and solve lots of problems. But it's also important that if you design something that it actually does something useful and that you actually prove that it can do what you claim it can do and that you get to that endpoint. So you want to get to where you're actually helping patients as soon as possible so that you can have a major impact. And if we go based on the background into our Everest expedition, we'll just try to carry that engineering forward. Our aim was to compare altitude-induced pathophysiology. So as people go up in elevation, they have less oxygen. Their bodies tend to start breaking down. They start having cognitive deficits. They start to look like cardiac failure patients. And so our goal was to look at the effects of going up a really high mountain at basically starving yourself of oxygen and comparing it how it looks to some of our clinical patients. The SPPDG was given 11 weeks to develop from scratch a remote monitor to field for the North Face National Geographic and Mayo Clinic sponsored Mount Everest expedition. And the device had to be rugged, had to run 24/7, completely autonomously, had to have high quality data. And this was pretty fun goals, and 11 weeks, and everybody wants to get their device on Everest, and so the team really dug down deep and worked a lot of overtime and actually generated what I'll show you on in 2 slides called the Mount Everest device. And so it was this small package shown in this was Conrad Anker. He climbed Everest that expedition wearing the device without oxygen. But they were wearing this device, and we had 2 leads because the feedback from the climbers was that they didn't want more patches because they weren't going to be showering for weeks on end, and they would be wearing the same clothes. And so we worked iteratively to find something with them that met their expectations. They wanted thinner, not smaller and thicker, and so there were a number of trade-offs. And so you can see that we had 2 leads attached, and if you can't see in the photo, they're essentially located here because when you bend and you move with a pack or climbing, this part really doesn't have much motion artifact, which affects your signal quality. And so we found experimenting in doing engineering experiments in a lab that was a good location where you got great ECG signal but not a lot of artifact. And also by placing the device there, you can get an idea of posture. And so you can begin to extract more information than you would have if you located it maybe on the belt. So you can tell what the central body is doing. And then the Mayo team also was kind enough to go along and support the climbers and take the devices. So they would run for 2 weeks, but it was a 6-week climb. So we changed devices 3 times on them. And we fielded 38 devices, and all of them functioned, and all of them came back with what I would claim is high quality data. And behind that was the SPVDG team. And again, to get this device up to Everest, it was an incredible effort, and a great number of people made significant contributions. In addition, we've had a number of generation of devices over the years moving forward based on this Everest and looking back on activity-only monitors. You can get a lot of information about somebody's wellness by just looking at how much activity they have in their life, and especially in kids. There's been a long theory, have them standing during class, have them constantly moving during school, and obesity will go down. And this is what the actual device looked like. We won't go into any of the parts here, but it was just a very, I would say, simple board with some fairly tight tolerances to get it packed into that little package. And ultimately, what was the problem was not the components, but its power delivery. And so we spent an incredible amount of time getting every single operation on the device effective. And since the Everest expedition, this is where the most effort has actually been applied. And with the same device, we doubled the battery life, or the effective run time, with a 2.5 times sampling rate. So an incredible increase in data rate with the exact same device, just by improving our firmware. And if anyone doesn't know what firmware is what's running on the device as opposed to software on your computer. So if I say that, you'll know what I'm saying. But you can see how every single piece of energy is expended in the device. And I actually spent an incredible amount of time doing calibration, and that's what I kind of wanted to highlight here. Is just getting the device is a huge effort, but then demonstrating that your device is actually recording what you think it is, and that it's repeatable and it's accurate, is, I would say, 80% of the effort. And making people, or helping people, believe that you're actually measuring what you think you're measuring is also another, we'll just say, an adder. So this is what the accelerometers on the device would see, is if you put them on a table that's been precision leveled and is on concrete foundation that actually goes down to bedrock, so it's stable as we could get, and then you level it with the machinist level, so it's as level across the whole table as you can get, and you put your device flat on it, and that's been machined to some NIST, National Institute on Standards and Technologies, tolerances. You can assume that device will read true every single time you put it down on there, and then you begin to put shims underneath of it, or small pieces of metal that are extremely thin, and you can begin to see the device tilt. And so we built up a number of automated tests to demonstrate that we could rapidly calibrate this device, and that you could begin to see the individual measurements, how they break down. So if somebody says they can tell you where a device is or what angle it's at, you would want to see, well, what's the probability of error in their measurement? And so we did that for every single one of these devices, going back and building off of what Dr. Wood did in 1938, and building up distributions to demonstrate that you could see a difference between a 0.2 degree angle or even smaller. And so I think this is one of the major steps we took to make our measurements repeatable and believable. Another thing we did was, if people are walking, at what frequency are they walking? If their arms are swinging, at what frequency are their arms swinging? If somebody has a Parkinson's tremor, at what frequency is that tremor? And how do you actually prove that they have a tremor? And so we took it another step and we started looking at frequency analysis to demonstrate that our device was stable across all the frequencies of interest. We demonstrated, and then it snowballs, as I mentioned. You can get a device, but then to calibrate, you have to have a reference standard, basically to calibrate your reference. And so you have to have a laser interferometer that has accuracy much greater than the device that you're using to calibrate. And so all of this played out to show that, in fact, we were getting a 0.02 G plus or minus acceleration at 5 hertz or 10 hertz or 20 hertz. And you could do this across your frequency of response. And then the plot that I really love is you have a device in the clinic and it sits on the clinic forever, and then somebody puts it on. How much air do you have? If you've never done the study to demonstrate that your device is stable through time, sitting in a package, then you don't really know. And what's worse is you don't know if it's significant to the measurement that you want to make. And so we were very fortunate that we had a clinical collaborator who was doing an orthopedic study, and they fielded our device over a number of years. And so every time they would field it, we'd bring it back in. We'd do that really quick calibration of the device and archive the calibration file, and demonstrated that as your devices get further away from their calibration point, there's intrinsic drift, and then you could decide based on that drift whether or not it was calibrated enough for your experiment. But the error clearly grows if you do a calibration and run the experiment, and immediately there's very few outliers in the experiment. But if you wait up to 818 days between calibration, the device has drifted. And so it may not be significant, but we were able to do the measurements and demonstrate the expected error. And as you look at ECG, how well are you able to reproduce a measurement of electrical activity in the heart? And so we did do a lot of testing to demonstrate when we said we had 60 beats per minute or 88 beats per minute. It really came back at 88 beats per minute. And then the fun started, which is we actually got to field these on the athletes. The extreme athletes came to Mayo. They did their baselines, and so we put the devices on them, and we verified that our devices, before we sent them to Mount Everest, would record across all the mountain climbers, and that we got useful, which I would call fairly low noise, very high quality ECG traces, even though we're only doing what's called one lead or a single lead pair ECG. So we were able to get good noise rejection and demonstrate the devices worked. And then they went up the mountain and they wore these devices, and it was really fun to get the data back. So this is what about an hour of data looks like, and it's really quite useless to look at it. And so you start zooming in, and the question is really, where do you zoom in? And so you look for where there's a big change. And what you actually see is a pathophysiology. As the person starts breathing faster, their heart starts beating faster because they're hypoxic, and then they catch up, and then carbon dioxide gets off-gassed, and they go hypocapnic, and they stop breathing, and they slow down, and they were miserable. And this trace essentially shows that. And so what you see at this jump here is they had an ectopic or missed heartbeat, which is a pretty significant pathophysiology. And it's buried, this little needle, in the haystack here. And what it also corresponds, if you start aggregating data across sensors, is they were in a particular position, lying on their stomach, tilted to the left. They missed a heartbeat. They just decided to roll over at that point, and they moved to a different position. And you could just go through the data and read out how they were laying, how uncomfortable they were, how poor their sleep was from these traces. And then you can begin to look at what it looked like when they were under stress. And I think what was really interesting, we generated so much data, there was basically no way to look at it with the methods we had at the time. And so when we do a deep dive at the end of the Everest expedition, we can really summarize all the devices worked, the data was of high quality, there was excellent compliance with the climbers because we took into consideration what they wanted in a device. We gained a lot of knowledge, skills, and experience. We received additional useful feedback. Our firmware improvements have dramatically improved the device, but the post-analysis was manual. If we get over here where our opportunities are to improve, this is a really terrible idea, but it was all we had at the time. And when you have 96 gigs of data come back and somebody asks you how good it is, and you start streaming through it, you get a good assessment, but it's not perfect, it's not objective. It doesn't tell you across devices were all the devices equally stable. Was a particular person more sick than another person? And so I think this has really opened up a whole another area of research between Dr. Holmes and myself and a number of other collaborators, is really trying to advance automated analysis and objective assessment tools. And so as we move forward, we're trying to automate everything so that I don't have to sit there and screen through it, or David doesn't have to do that. And we're trying to distill what in there was clinically useful. So somebody rolling over, probably not clinically useful. It's a great chart. But what does that mean for people's health as we move forward? And so we're looking at, as is commonly used in the press, this term biomarkers. And also what we found is that it was very hard to scale our device in the fashion that it was built, because it was built so rapidly. And so we've been developing a lot of methods to accelerate device development, design, numerical validation, and design optimization, so that when we actually generate a device, we have a good idea that it's going to work, as opposed to we built the device and we're not quite sure what its performance levels will be. And so this, I'll just cover the first two. So we had all kinds of data. You can go in and you can extract these things and get terms like heart rate variability and what does that mean. I would claim it's not a systematic study, so we really don't know, but it looks, again, like a pretty chart. And if we look at a Parkinson's patient, we can go in and we can see why. We spent all that time calibrating on frequency harmonics, because you see harmonics in tremors, which, if you know physiology, it's the loop between the muscle or the tendon and the central nervous system and the local nervous systems and your reflexes, and all these things matter. And then how does a drug affect it? Can we make an objective measure of drug performance, of drug dosage, of drug timing? All these things are very interesting, but unless you have a tool that you've actually calibrated, really calibrated, you don't have an objective measure. And so I just want to define for you what I saw this statement of what a biomarker is. It's an indication of a medical state observed non-invasively from a patient that can be accurately and reproducibly measured and is predictive of a relevant clinical endpoint. And so I would claim the last two slides are not predictive of a relevant clinical endpoint, so they're features of the data as opposed to a biomarker. And so I think it's very important when you start becoming young engineers and you start moving out into the world and trying to solve huge problems that you consider fundamentally what is the definition or what is a good description of what you're trying to solve, and then go back and figure out if that's really relevant. And so this leads into all the research we've been doing since the expedition, which is led up to classifying data in a very efficient fashion so you can run this on the device, that small Everest unit. Is it good quality data or is it poorer quality data? If you can't tell, you don't know how much time or power to spend on the device processing data. You don't know if you should send that data somewhere. You don't know how much credibility to give to that data if you're going to make a clinical decision. Should you be calling an ambulance for a patient? Well, if you have really poor data, probably you should put a little header in there that says the data is really poor, but it looks like, you know, they're flatline. Well, they're probably not wearing the device. But you should look at the history of the device. You should look how you got to there. You should look at other variables at that point, and I think this is what we're really looking at. So this is essentially the classifier. So on the left, we have a poorer quality data. So the red line is the ECG trace, and we've generated data here that we understood the amount of noise we've put in, and so we see that this step function is or stair step is our desired data quality, and that our metrics actually show within reason that we're able to determine the quality of data in a very efficient fashion that can run on the device. And with that, you can start to make decisions on an extremely low power, extremely computationally limited remote monitor. And we were only able to make those thresholds because we went out and we did a deep dive into the individual components of a physiologic or ECG waveform. So you have all these little components. Every time your heart beats, you have P on, P off, QRS on. All these exist every day in your body. And you want to know, if you're trying to make a measurement, how good does the data have to be to pick out a particular waveform? So if you want to pick out the big component, this QRS component, you can have pretty poor data and still pick it out. If we go back, you can see it here. This noise is terrible. This is terrible, but you can still statistically pick out the R wave. As you start getting into cleaner data, you see that R wave is very well resolved, but it's very tolerant to noise. So you should know this when you're looking at features. And so you map out what is your SNR tolerance or data quality tolerance for every single one of these metrics, and that tells you how good your device has to be. And then, if you have all that, then you can actually write the code and shove it back on the device and demonstrate that you get the same effective assessment of physiology on a microminiature platform or an MSP430 or something weaker than your cell phone that has the same sensitivity and specificity or quality as something running on a huge supercomputer. And that was because we tailored it to the specific physiologic waveform or feature we were interested in, and we spent a lot of time optimizing code. And what's interesting is, with all this processing running on that little Everest device from 2012, with doubling the power consumption, we're able to add all this processing to that MSP430. And now you can begin to aggregate data and look across lots of waveforms. And I think this is where all the future engineers will have lots of fun, because there's going to be so many more sensors, and you're going to be able to use machine learning and artificial intelligence and all the fun words to attack these problems. But you have to consider where your data comes from and how good the quality is. And so in this case, what we would see is that we know if somebody's standing. So if you place the device on their core, exactly how we showed in the Everest, you can look at how they're standing. If they're standing, supine, left side prone, right side walking, etc. We can see when they're moving to change positions. In this case, in an experiment, we can see that signal quality decreases as people are moving more. And then we can see that their heart rate also increases as they begin to change posture. And a lot of this is physiologic. If you're laying down and you stand up, you have a great orthopedic, or I can't remember the term, but you have a great response to standing up. And if you do it too quick, some people will lose consciousness, but you can see all of these effects in your aggregated data. And so if we conclude just the device section, I think you really need to look at not just developing something by yourself, but aggregating a strong team across a number of different disciplines or different interests so that people have different perspectives. You should aim for something that actually addresses a significant financial or human burden on society. That's the Mayo Clinic, or my personal take on it. And you should allow for rapid integration into either everyday life or the clinical practice. And I had just a brief subset of slides as we look at the future engineers. I think you'll want to do a lot of research on the background, and that's where I spend a lot of my time, just reading articles, reading books, so that when I go to define my goals or my expectations, I have an idea if it's new, if it's novel, if it's useful, and or what I can learn from other past experiments. You then want to find very relevant articles or references, and this takes a lot of time, but the next one will kind of help work with your parents and teachers to identify experts, such as a lot of the people who are very esteemed and sitting out here in the crowd, to help you scope your frame of reference so you can go out and find a right book to go get from the library to help you tackle the electronics problem that you're interested. If it's regenerative medicine, maybe it's a recent paper in a journal that you don't have access to, but somebody else can help you get it. I think that's very important. I think it's also important to look at rapid prototyping, whether it's code or it's devices. So you should pick a framework that's widely available, widely used in the field of study. So if 10,000 people are using a type of of rapid prototyper or a piece of code or language in a field and you choose something completely different, they might know something. Maybe there's a lot of libraries that are available that will help you develop more quickly. Maybe there's already calibrated or ground truth information out there that you can accelerate on. And you also want to document your work. And that's very painful, and people are laughing because basically every single line of code should have a reference or a description of why you're doing it. And if you don't know why, you should probably rewrite that line of code 10 times. So, and I also want you to know, I chose one way to do it, Earl Wood chose another way, Dr. Gilbert chose another way. There's so many ways to do research, and if anyone tells you there's only one way, they're really wrong. It's their way. It doesn't have to be your way. So this is a great example. This is a nice paper that came out very recently. They took 7 different devices, completely unrelated to each other. They put them on a whole ton of people, and they let people go about their daily life. Then at the end, they recorded their data or collected the data, and then they did blind machine learning on it to see what features they could extract versus their clinical outcomes. And they determined that they could detect Lyme disease early, which was not what they primarily wanted. But it was of interest because it was one of the first studies, and it shows that the NIH and other organizations are very interested in it. And I think, you know, this is really interesting because what it allows you to do is extract out what are the features that are interesting. So there are a lot of people very interested in Lyme disease, and they could determine which physiologic variables could target early detection of Lyme disease in somewhere such as Wisconsin, where it's incredibly prevalent, or out east, where it's also very prevalent. And I think that's very important. On the other hand, there's groups in the Army who are very interested in people dying from blood loss. This is very interesting in trauma. So they did a completely different type of study where they built the most incredible physiologic model possible, where they essentially demonstrated that blood loss was exactly equal to pulling a vacuum on people on the lower body. So you pull blood, exactly what was happening with G-induced loss of consciousness in the pilots. You pull lower body negative pressure, relative negative pressure, and you demonstrate that it correlates with blood loss. And so this was an example. This was incredibly expensive. It's taken 20 years, and it's on the complete opposite end of the spectrum, but it's very well regulated and controlled. And then I think you also have to know that there's lots of things outside of your specific little field that might help you validate your measurements. And so you should consider that validation and calibration, as I mentioned, are 80% of the problem often, and that you should look outside of the box to find solutions to that. You should also define tolerances that are good enough. So if you want to get something out into the field, you probably don't have to take it to the nth degree up front, as we did for every single measurement, but we knew what we wanted to measure, so we did do a lot of that engineering. If we were just trying to get something first proof of light in an experiment in the clinic, we could have probably gone with an earlier version that didn't have so much validation and wait while the validation was ramping up. And then also consider that there's an incredible number of people that have to contribute to do big projects, and I'm very happy that a number of them are here today as members of IEEE. And so I appreciate any questions that people might have.
Questions from the audience?
Where did you build the devices?
So we fabricated we did not fabricate the boards. We had the boards done by a standard PCB manufacturer, but we assembled everything in-house. And one of the problems is we had very tight tolerances, and so a lot of the parts were hand pick and placed. Sure.
Did you say that some of the climbers actually helped specify what data was to be gathered?
They specified what they would tolerate. And so everybody wanted a lot more sensors, but there wasn't time to integrate and calibrate and validate a lot of sensors. We'd love to have some sort of measure of blood oxygen saturation. We'd love to have a direct measure of respiration, body temperature, barometer, all those things. A lot of it was limited by battery power. So the altimeters that were available in 2011, 2012 were too power hungry, and they themselves would have consumed more power than the entire rest of the system, so we left them off. But there's lots of, so they said, we want thin, we want it on the chest, it doesn't interfere with our straps, we want 2 sticky patches, we're not going to do 3, and they were very tolerant.
So I know you mentioned that there might be a future where it's integrated into daily life and monitoring just people in public. Do you see that running up against any kind of patient privacy?
Well, I think people would want to have their own power to say yes or no. If they came into a physician's office and the physician said, I'd like you to wear this device because we'd like to assess your cardiovascular state, and it's no different than other devices that have been out there for a long time. However, we can get a better picture of across more variables. I think that would be their choice. I think you would never put a device on somebody without their consent, and that's the most important thing. So that's the fundamental basis of medicine and experiments. So there's maybe you know of the institutional review board. So if you were going to do an experiment, you would have to go up and have it reviewed, and they would assess whether or not it's useful. If you were going to do it more clinically, you would go up to the FDA, and then you would state to the FDA your case, and they would also help you make that decision. And then the physician ideally would prescribe it.
So, Clifton, you did an amazing job getting across the idea of calibration, validation, reliability, and how important that is in any kind of equipment like this. So we all have these in our pockets. So this is a year-old iPhone 7 Plus, and it has an accelerometer, a magnetometer, a gyroscope, and we use this for measurements like that, like activity, just something simple. Or we have our watch, or we have our other, strap-on accelerometer. What do you think the chances are that these are giving us valid, reliable data?
Well, I can tell you we've looked at a number of devices that have been on the market, and I suspect you have as well. The repeatability is often very low, but what I would love to see is if you're going to use a device that you take an initial measurement, you can do it on a simple table. If you have some measurements, you rotate it through 6 degrees, and you see if it lines up with what you expect. It should go 1, 1, negative 1, 1, negative 1, and you should be able to calibrate and do a simple calibration. So I think there's lots of devices out there, but I would personally test them all before I used them and before I put any faith into the measurements I was taking. And I would certainly put error bounds on it.
What is the acceptable error bound you all determine for your device?
Ours was, I believe, ridiculously low. It was as good as the precision gauges we could make. But as you saw in that plot, it's a histogram, so there's noise in your data. Devices aren't perfect. So if you take a 5-second average at a particular really small angle, you could see it because we're collecting, say, 100 samples per second. So you have 600 samples. You get a nice distribution. They were nicely distributed, so you could get your mean. But if you didn't know that your mean, you didn't know that you had, let's say, normalized data or data that's distributed about a particular number, and it was in fact tailed, you would never know because you hadn't made the measurement. And so that was what we were doing. And I think, again, you can use any device, but you should make sure that you actually verify that it's giving you what you expect.
Yes, sir. Do you need off-body sensors? Like, so I'm thinking of somebody who just happens to live in let's say there's a building being constructed next door, so there's a lot of vibration. And if you just had a sensor on the body, you might think that there was something strange going on.
So we do not at this time, but I would guess if there was a concern that you could put a second device stationary near them, but we hadn't addressed that at this point.
So you're 13 or you're 15, and you're here and you're listening and you're thinking, I want to be an engineer someday, where do you start?
Well, you graduate high school is the first, most important thing. And you listen to your parents.
That's an excellent answer.
Yeah. So I think it's really, you got to have a curiosity. You got to be willing to find something that excites you. You got to have the conviction or whatever you want to call it to actually follow through with something. So you say, man, I really hate tying my shoes. I'm going to invent something to tie my shoes for me every morning, and it's going to be super simple and super awesome, and I think I know how to do it. Well, okay, that's a great idea. Why don't you write it down? Now start formulating what it is that you're actually trying to address and why. And then you begin to slowly build upon that I mean, this is a great question. You build upon that simple concept, what it is you want to do, and you start to formulate why it is you want to do that, how you're going to do it, how it's going to change people's lives. And as you progress through that, you'll eventually realize that you've basically formulated your plan of attack. Now you have a plan of attack. Start finding references out there. Has somebody already done it? That's a great search, the obviousness search. This is exactly what the U.S. Patent and Trade Office does when you submit something. Has somebody already done it, or can I combine two things to get what you've done? And now that you've done that, you begin to build up concepts of how you attack it. So first, I need to have a sketch, or I have some operations that I go through. First, I loop it over, and then there's a rabbit, and then it comes through. But, you begin to build this up, and it's a very, tying your shoe automatically. That's a great problem. I think there's a patent from Nike on that. I'm pretty sure the self-tying shoe. But that's how I would start. You have to have it. You have to formulate, write it down. What is your idea? And review it with someone you trust. Go to your parents. I think that's that innate curiosity. You want to do something. And you actually start taking steps. Once you get past those first few steps, maybe that didn't work out. So find something else you're interested in. Automatic. I don't know. Everything's automatic. Find something else. Automatic waffle maker. That's what I want. So you formulate that. I think you graduate high school. You've done extremely well. You pick a program that relates to something that you're interested in, and I think that will be the second thing. Really focusing in college. You'll have lots of time to take lots of classes, and the advice I always got was volunteer in labs. So professors have very few dollars, and those dollars go further if students go in and do stuff to help them. So my professor, June B. Park at the University of Iowa, said volunteer to wash dishes in a lab, and eventually they'll see value, and they'll start giving you advice, and they'll start helping you. So that's another one. And then, of course, you can go anywhere once you have your college degree. Whether you come to Mayo, you go directly into industry, you start contributing immediately as an engineer. You go to medical school, you go to, art. Maybe you want to make violins. That's fantastic. Now you know how a harmonic works. What is a harmonic? A single wave, and it's really hard. And people have been trying for centuries to make perfect violins. Maybe you'll have the new perspective.
Very good.
Yes?
So I want to make a shout out. So, we talked about things you can do. A great place to start is Mayo has a high school mentorship program to get students in your position into labs. And we have a representative from that program, Alex Weston. So if there's any interest in more information about that, Alex can answer your questions. But that's a great place to start. I started my research just volunteering undergrad lab at my university.
Excellent. Let's thank our speaker.